it's probably kind of hard to tell on the video, but most of the colonies on this plate are blue and some of them are white. And so each of these colonies is like a glumpy dot thing, and it represents a genetically identical bacterial clump. So basically the bacteria, one bacteria gets settled in and it multiplies and multiplies on top of each other so you get this clumpy thing. The reason some of them are blue and some of them are white is because it's using this technique called blue-white screening, which is an easy way to see if the bacteria probably have the gene that I put in them, tried to put in them. And basically how it works is that when you put the gene, you do it so that you put the gene interrupting, so you put it in the, insert your gene in the middle of the gene that allows the bacteria to make this blue product from a chemical called X-gal or blue gal that you put in the plate. And so if the gene you want to put in gets inserted, it's going to disrupt that enzyme beta-galactosidase that the bacteria use to make the blue color, and so you won't get the blue. So I want the white colonies, and they probably have what I'm looking for, but I'll have to do sequencing if I wanted to actually confirm. In molecular cloning, we can take the genetic recipe for a protein and stick it into a circular piece of DNA called a plasmid. This plasmid will serve as a vector or vehicle for getting that genetic recipe into bacterial cells to get the bacteria to make it for you. We want to make sure that only the bacteria that actually take in that plasmid are able to grow when we feed the bacteria, and so we do something called antibiotic selection. So along with our insert, so the gene we put in that plasmid is the antibiotic resistance gene. And if we grow the bacteria in the presence of the corresponding antibiotic, only bacteria that actually have the plasmid will be able to grow. But the question remains, do those bacteria actually have the insert in that plasmid? So by insert, I mean the gene or the modified form of the gene. Only real way to know if the insert is there and is all properly spelled and no typos or anything is with DNA sequencing. But you don't want to waste your time sending off a bunch of clones for sequencing, colonies for sequencing, if they don't actually have your insert. So there are quick checks that you can do to try to see if it's in there, and these include colony PCR, which we looked at the other day, where you make copies of the region containing the insert and see if it's there, or you can also do cut sites. So those will give you like rough ideas of the sizes of the products, but it won't actually tell you if the product is correctly spelled. And the key drawback of these methods is that you actually have to put in some work after the colonies. So you have to like pick the colonies and then do some work on them. So either colony PCR, you can just work directly from the colony, or with restriction digest, you have to actually purify out the plasmid first and stuff. So basically, these take a little bit of effort. But what if you could use a screening method to be able to just visually look at the colony and see whether it probably has your insert? And so this is the idea with blue-white screening. So the word screening, so before we had selection, so the antibiotic is selecting, so it's only allowing the bacteria with the plasmid to grow, and and weeding out the others. With screening, however, we're not weeding out the bacteria without the insert. We're just showing you which ones don't have the insert. So you're kind of like flagging the bad ones and the good ones. You still have the antibiotic selection going on, or else you just get like a whole lawn of bacteria. So you have these both at the same time. So you have the antibiotic to tell you that the plasmid is there, and the screening to tell you that the insert is probably there. So we can't tell you for sure, but it tells you that something has disrupted this beta-galactosidase gene. So where the heck did that come from? So beta-galactosidase is an enzyme, so a protein, a reaction speeder upper that bacteria use to hydrolyze or use water to split this sugar called lactose into galactose and glucose. These, which are soluble, but they don't have conjugation. So conjugation is this weird thing where atoms form molecules by sharing pairs of negatively charged subatomic particles called electrons, and in the process of conjugation, it's kind of like a communal electron sharing. And the reason why that matters is because it allows them to absorb light in the visible range. But these molecules don't have conjugation, so we can't see them. So that's not very useful for us because we can't tell if it wouldn't serve as a good marker. But it turns out that beta-galactosidase only really cares about that galactose part, so we can trick it into cleaving things that do make a color. So for example, X-gal, when it gets cleaved by beta-galactosidase, you get this extensively conjugated product that absorbs light and lets us see it. It's also insoluble because it doesn't have those OHs, so it precipitates out. And this is what you can see in those colonies. You get those, like, that blue precipitate builds up and you see the blue. There are also other colored versions. So beta-galactosidase functions as a tetramer. So 4 units, so it's 4 copies of the beta-galactosidase subunit. So the monomers are the individual subunits, and then they dimerize, so they pair up, and then that dimer dimerizes, so you get a tetramer, so 4 of these together. The important thing about this is that only the tetramer is active, and the tetramer, making the tetramer requires this part of the beta-galactosidase unit called the alpha peptide, which is on one of the ends. So I'm showing you this like green thing. With blue-white screening, we tie the production of beta-galactosidase, as indicated by the formation of a colored product, to the presence or absence of an insert in the vector. So as we saw, you need that alpha peptide in order to form those functional tetramers. But the bacteria that we use have a mutated version of this, the LAC-Z gene, which has the instructions for beta-gal. And so this mutated version is missing the part that makes the alpha peptide. So it can't form those functional tetramers. However, you can provide that alpha peptide fragment on a separate gene. And you can do that on the plasmid, what you want to put your insert into. And you can then, what you do, so if you have an empty vector, that alpha peptide gene is undisrupted, so you can make the functional beta-gal. However, if your insert gets inserted, so the gene that you want to put gets inserted into the alpha peptide gene, then you can't get that alpha peptide made, so you can't get that functional product. Without that functional product, you cannot convert the X-gal or whatever variant you have into the colored product. So the white colonies have your insert because they can't make the beta-gal to make the colored product, but the blue colonies just have the empty vector. And so this allows you a quick look to see whether the insert you have tried to put in is actually in there. The bacteria won't make beta-galactosidase unless there's they think they need it normally. So normally they know how they need it because there's lactose present, and one of the things beta-galactosidase can do is turn lactose into this form called allolactose. The lac promoter is in front of the LAC-Z gene, which codes for the beta-galactosidase. And so there's this protein called the lac repressor that sits on top of that promoter sequence and prevents the beta-gal from being made. However, when this allolactose compound is present, it'll bind to that repressor and de-repress it, so make the repressor fall off so that you get expression of beta-gal. So it turns out that allolactose isn't very so this allows us to get the bacteria to make beta-galactosidase even when they wouldn't usually. If we were to add so this allows a way to get bacteria to make beta-galactosidase even if there isn't a need for it for lactose. So you might think, why don't you just add allolactose? Well, it turns out that beta-galactosidase can also split allolactose, which wouldn't be very helpful because we'd keep having to add it. But there's this molecule called IPTG, which mimics the allolactose and can bind and de-repress the repressor in the same way. And so this allows for expression of beta-galactosidase. So what you can do is you can put IPTG and your X-gal or blue-gal or whatever substrate product you want into the bacterial media along with the antibiotic. So when you're making these blue-white screening plates, you have the antibiotic, you have the IPTG, and you have the product you want to convert to a colored product. So this whole repressor thing, IPTG thing, might seem familiar to you because we talked about it with recombinant protein expression, where we put the lac repressor in front of the gene for a protein, for the T7 polymerase protein, so that D7 polymerase gets made, and that allows, and then you have a T7 promoter in front of the gene for your protein, and so the T7 polymerase, when it's made, can then get the cells to make your protein. And so this is just another use of IPTG. It's the same same system we're talking about, except here we're using it in a different way.